All right. Well, welcome everybody to College of Science virtual seminar for the second week of November already. Oh my gosh, semester is going very, very fast. Anyway, it's a pleasure today to have our own Associate Dean for Research, Miri VanHoven, as our speaker. She's definitely upping the ante here. She's been bugging me to give a talk for a while now, and I'm going to do it, I promise. It'll be a little while. Let me give you a little introduction to Miri. So as I said, Miri is the Associate Dean for Research in the College of Science. She earned her bachelor's degree in biology.
Molecular biology.
Molecular biology. I knew it was more than just biology. At Pomona College down in Southern California. She got her PhD at UCSF up this way from where I'm sitting, and then did a postdoc at Stanford University before joining us at San Jose State in 2008.
Yes, very good.
Since then, she's been an assistant associate and full professor in the Department of Biological Sciences. And for the last couple of years, both the interim and actual Associate Dean for Research. And she somehow manages, amidst all of her administrative managerial duties, to also keep her lab going with a whole bunch of students. And that's what she's going to tell us about today. Her research area is the formation and establishment of nervous systems. And we're going to get to hear all about that in just a couple of minutes. Her work's been supported by grants from NSF and 6 NIH research grants. Her work has been published in top journals with many of her students as co-authors, as well as faculty in the College of Science and collaborators from UCSF, Brandeis, Vanderbilt, Yale, to name just a few. Today's talk is entitled Genetics and Activity Shape in the Developing Neural Circuit. And it's all about worms, and I'm very, very excited. As usual, go ahead and put your questions in the chat or the Q&A. I'll interrupt Miri if it looks like it's worth doing so at that moment. Otherwise, I'll give you the chance to ask your own question at the end of the talk.
Definitely feel free to interrupt me. Totally fine.
Take it away, Miri.
All right, let me make sure I have the right thing up. Okay, are you seeing this okay?
Yes, it's all good.
Wonderful. Okay. So thanks so much for the introduction, Michael. Yeah, one of our junior faculty had to cancel last minute because of an experiment that had to be done, required weather conditions and everything, and collaborators. And I thought that it would be a good, good experience to make myself give a talk. So I stepped in, but I apologize because I've tried to push it into my schedule. So if we discover along the way that anything's incomplete, I sincerely apologize in advance. So I'm going to tell you today about how both genetics and activity shape the developing neural circuit. And this is probably the broadest audience I've ever given a talk to. So definitely feel free to break in with any questions you have. There we go. So I'm going to talk about the nervous system. And the reason that I am so excited about studying the nervous system is that it's literally responsible for everything that makes you you. It's required for perception, for thought, for behavior. If you do it, it's because the nervous system made it happen in almost all circumstances at some level. So I'm really fascinated by how such a complex system actually forms. And the nervous system is huge. The central nervous system alone is composed of 100 billion cells connected through trillions of connections that we call synapses. And I'll tell you more about them in a little bit. We typically, for those of you who are biologists and have seen pictures of neurons, we typically imagine them alone out in the middle of a vast, vacuum plane finding a single partner and forming a connection. However, that's not really how it happens. The vast majority of the nervous system is densely packed, like you see here in this beautiful micrograph from Livet et al., where they actually show all of the individual neurons in a tiny region of the cerebellum, which is this part of the brain right here. You can see these are all cell bodies like here and here and here and here and here and here. And they're all kind of just jam-packed. All of these little bodies are cell bodies. All of these little circles are, and all these long extensions are their neurites, these long cellular extensions that they grow so that they can communicate with each other. And they're just jam-packed into these tight, compact areas. And yet they form this just elaborate and extensive and absolutely correct system of neural circuits in almost all people. Of course, when they don't form them correctly, we get neurological disorders, things like schizophrenia, like mental retardation, that cause serious problems to the affected people and their families. And so the other allure of studying the nervous system development is to really understand it at such a deep level that we might be able to ameliorate some of these neurological disorders that arise when development goes wrong. So today, so the nervous system forms in multiple steps. And actually our lab has, and many, many others have studied multiple steps in neural development. And I'll tell you about some of them now. So the first step, actually even before these genetic signals initiate synaptogenesis, is neural migration. So the cell bodies have to actually migrate to the appropriate place in the body. And then they extend these long neurites that we call axons and dendrites. Dendrites that receive the information and axons that pass them on to the next neuron. And I've just drawn one neuron here, but you can see as it extends into this target region, even once it reaches that region, it still has multiple potential partners, multiple cells that it could choose to form a synapse with. And yet they reliably recognize the correct synaptic partner and form synapses. And these synapses, so here I've drawn one neuron here in red, one neuron here in blue, and all these other incorrect partners in gray. And then this little green dot here is a synaptic connection, or probably honestly a cluster of them, as I'll tell you in a little bit. So this first process by which neurons actually recognize the correct partner within kind of a sea of potential partners, we call recognition and synaptogenesis. And the first part of my talk will be about that. That's really the nature in the development of neural circuits. However, soon another equally interesting process is layered on top of this, and that is activity. So activity through the nervous system, and specifically through those circuits, can affect how many synapses are formed with that correct partner that's already been recognized. And so this activity-dependent post-embryonic process can help each individual animal form circuits that are at the correct synaptic level to help them respond appropriately to their environment. And so I'm really interested in studying both these processes in the same circuit and understanding how they work together to ensure individuals that can respond to their environment. So let me tell you a little bit more about these connections between neurons. The connections between neurons are called synapses. There are other kinds, but these are by far the most plentiful and impactful, I think. And the way that they regulate communication between the neurons is that, and we've just really zoomed in. I should have just said this before, but if you look at this see this little box here, like that's what we're going to zoom in on now. So now we're just looking at one synapse, and you can see this is what we call the presynaptic neuron, the neuron that is going to communicate to the other neuron. And this is the postsynaptic neuron, the neuron that's going to receive information. And what the presynaptic neuron has, the actual molecules that communicate are called neurotransmitters. And you guys have probably seen these, if nothing else, in annoying commercials for various drugs that you can take. So on the bright side, people now know what neurotransmitters are. That wasn't always the case for me. But you have these vesicles, these membrane-bound circles in the presynaptic terminal that have these little molecules. The molecules are actually much smaller than this, but if you draw much smaller, nobody can see. And what happens is when one neuron is stimulated or depolarized is what we call it, but every time I say depolarized, you can just think stimulated. They release the neurotransmitter into this space between the two neurons, which is called the synaptic cleft. And then receptors that are proteins that are transmembrane proteins in the postsynaptic neuron, or the neuron that receives the signal, are bound, and that affects the excitability of the postsynaptic neuron. So if it's an excitatory synapse, that will excite this neuron, and then it'll send a signal all the way down it and communicate with its next partner, and so on. Okay, and this is how the nervous system does everything. It sends signals through a relay of pre-selected neurons in a circuit that finally accomplishes its goal. So what do we know already about mediating this recognition event and this initial synaptogenesis between two correct partners? Well, people have studied this in a lot of systems, and they have found several families of proteins that can initiate this synaptogenesis. So they're the cadherin family, which are these transmembrane proteins. This is the membrane here. These are single pass transmembrane proteins that have domains called cadherin domains and other domains that are just little sections of the protein that are conserved that help them interact transsynaptically or across the synapse with each other. Leucine-rich repeat proteins are another family of proteins that can mediate these interactions, very similar, except the extracellular interaction domains are called leucine-rich repeats. And immunoglobulin superfamily proteins, again, very similar, except the extracellular domains that help them communicate are called immunoglobulin and fibronectin type 3 domains. And this is a picture of how they can interact across. So if this is the presynaptic neuron, and this is the postsynaptic neuron, these are examples of how various protein pairs that can mediate synaptic recognition have been shown to interact. Okay. So we know a lot of kind of individual examples, a lot from mouse sections, some from cell culture, some from Drosophila, which is the fruit fly. What do we know about how activity regulates synapses? Well, the system we know the most about for this is called long-term potentiation, and it's in the hippocampus, which is an area of the brain that's involved in learning and memory. And this is really a picture that's aimed at the cell and molecular biologists to be satisfying to them, but to everyone else, essentially what is believed to happen. Decades of research have contributed to a model that I'm going to give a synopsis of in 10 seconds, so it will not be complete. But essentially, as activity goes through a circuit, you retain more neurotransmitter receptors at the postsynaptic site that is directly juxtaposed to the presynaptic neurotransmitter release site. And that makes these synapses more strong, and it can cause you to develop bigger synapses, and in ways that I won't talk about here, more synapses. But despite having studied this for decades, there are actually huge gaps in our knowledge. For example, we don't know how this happens in other areas of the brain with other neurotransmitters, even in and other neurotransmitter receptors. And we frequently don't know, we don't know even like which targets of these downstream kinases are the most important in many cases. So there's really quite a lot we still need to learn. So why don't we study humans? It's a good question, you know. I used to teach genetics. I had 180 students every semester. Why didn't I just grab some of them discreetly after the class, take them to lab and chop them up and start studying their brains? Well, first, I probably would have been fired, especially if I did it pre-tenure. But more important than that, or maybe less important than that, um, studying humans is really hard. We're really, really complex. And the things that we can do that are non-invasive frequently don't provide the level of granularity or molecular insight that we really need to understand these processes at the molecular level and to eventually develop therapeutics to address neurological disorders. And so what we need is a much more simple system, a system that we can easily visualize in animals that we really understand very well and that can be genetically manipulated easily. And it turns out that all of those things are amazingly, I'm not going to say simple because my students will yell at me, but can be done and done well in C. elegans. So C. elegans has a simple nervous system, 302 neurons compared to your 100 billion in the central nervous system alone. We know the synaptic connectivity of the entire organism, not true for any other model organism or any other organism at all. It turns out that the synaptic morphology, like the shape of the synapses, the structure, and the molecules at synapses are largely conserved. Looks like the nervous system essentially evolved once. And yes, it has gotten more and more complex as we move up into vertebrates, but that happens really in organization of the brain and not so much at this very basic level of synaptic connectivity and how the number of synapses are mediated by activity. Amazingly, they are transparent. This is not a section of a worm. We did not cut this into a tiny section and fix it. This is an entire live worm. So they are transparent. You can use cell-specific promoters to derive the expression of fluorescent proteins and label any cells you want almost. And you can just anesthetize. You literally can take the worms, take a slide, put a little agarose pad to make them comfortable, put a little buffer so they don't explode, and a little bit of anesthetic. You pick the worms into that, you put a cover slip on it, and 5 minutes later, you can look at the worms on a compound fluorescent microscope. And then you can even rescue them afterwards. You can take them off, remove that cover slip, put the worm back onto the plate, and it can live a long and happy and healthy life. So that lets us do a huge amount. And especially when you're studying connectivity, you really don't want to cut the worms up first if you can, or cut the animals up first if you can avoid it, because that can disrupt the connectivity. And we have the ability here to look at live intact systems and understand what's happening, which is just an unparalleled advantage of this organism. I could talk for the entire talk on what amazing model organisms they are, but I will not. I will move on at this point. At this point, I'll move into the main part of my talk, the first part, which is to talk about the genetic signals that initiate synaptogenesis with the correct partners. Sorry, I paused to read the chat for a second to see if anybody had a question. But it was just Walter remarking that it sounds like as long as she is using these worms, her data will be very transparent. Thank you, Walter. All right, so beyond studying C. elegans, we have focused on a circuit called the phasmid neuron circuit. So this is a schematic of a worm. And this is my favorite neuron in the world. It's called PHB. It is one of the phasmid neurons. There are 3 sets, PHA, PHB, and PHC. I like PHB the best, I'll explain. It forms synapses. So I'll explain its morphology in a second. This is its cell body. This is the dendrite that extends to this phasmid opening here where it actually can sense the environment, chemical cues. And then it extends an axon ventrally and then anteriorly into this region called the preanal ganglion, which actually then more anteriorly becomes the ventral nerve cord. And it forms synapses with this command interneuron here called AVA. I could tell you the reasoning of it, but I won't because it takes too long. It extends as one neurite through the worm brain, which is called the nerve ring, down to this ventral nerve cord, which is essentially analogous to our spinal cord. It goes all the way down the animal and all the way back here. And then the synapses are formed in an en passant manner along this region. En passant just means kind of along the axon there. And this is a blow up of that, because what we're really going to focus on most of the time is the synapses that form right here between the PHB and AVA neurons. Now, I want to tell you, I've only drawn PHB and AVA here, but that's not actually accurate. There are a bunch of other neurons. So here we traced an electron micrograph of a cross section that goes right across here. And you can see, so these are the PHB neurites here in red. These are the AVA neurites here in blue. These are the PVC neurites here in gray. That's actually the other primary postsynaptic partner of the PHB neurons. We won't focus on them today. And all these other gray neurons they're neurites that PHB could be confused by, could form synapses with, and yet they selectively will recognize AVAs and form the majority of their synapses with AVAs and PVCs. So it's a really great model for vertebrate systems that also have these really complex environments in which they faithfully recognize the correct partner. So if you're going to study synaptic connections between neurons, you need to be able to visualize them, right? You can't just look at them under a regular microscope, a light microscope. You wouldn't be able to see them, but you can light them up with fluorophores. And that's actually what I did during my postdoc in Kang Shen's lab at Stanford, was to design this novel marker for labeling specific synapses in live animals. And basically, we take a green fluorescent protein, which we call GFP, and we take a version of it. So if you don't know, green fluorescent protein is this amazing small protein that fluoresces green when you shine essentially UV light at it. You can, we got it from jellyfish originally, not me personally, but Nobel Prize winners. And then it's been evolved over time by scientists in labs to make it more stable, to do different versions. The version that I used was one that was called a split GFP. So GFP is a beta barrel structure, and there are 11 beta sheets. And what they did is separate out the first 10 from the last one. And it turns out that if they're separate, they do not fluoresce. However, if they come in close proximity, they will come together and they will reform the beta barrel and they will fluoresce. And so what I did was just to develop a flexible linker and link it to a synaptic protein called neuroligin, which is both found at presynaptic terminals and postsynaptic terminals or sites. And then I expressed that cell specifically in my neurons of interest, in this case, PHB and AVA. And we see in wild type animals, we see fluorescent dots or puncta that indicate that the synapses are there. And in mutants that I'll show you later, the neuroligins, if they don't form appropriate synapses between the two partners you're looking at, then the split GFP fragments do not come in contact, and they do not reform the GFP, and they do not fluoresce, okay? So here I'll show you a wild type animal. And these are actually the schematics that we've drawn, and now we've really zoomed in to just that area of overlap between the neurites. See the PHB neurites coming into that preanal ganglion, which is that posterior region of the ventral nerve cord. And then this is AVA coming in from the head. Okay, and so this is a schematic. This is what that actually looks like. right? With the neurites contacting each other and making this beautiful contact here. And you can see the what we call synaptic puncta, and these are the neuroligand grasp fluorescence, right? And I think I forgot to say, but we actually call it GRASP for GFP reconstitution across synaptic partners. This is an overlay. When you overlay green fluorescent and red fluorescence in a picture, you get yellow. So you can see that's right along this area of contact between the neurites. This, by the way, this super bright thing, don't look at it. It's the gut. We don't care. Actually, the gut is incredibly interesting. And a lot of people I know study it. So I completely take that back, but in the context of my talk, don't look at it. All right, so the other really cool thing about the PHB circuit is that we can have this circuit-specific behavioral assay that we can use to assess circuit function. And this was actually developed by a friend of mine, Massimo Hilliard, a long time ago. And essentially, PHB neurons in the tail sense noxious liquids. So I'm not kidding, he poured almost the whole Sigma catalog, maybe not the whole Sigma catalog, but a lot of it. He would just drop it at the tail and see if the worms responded. And it turned out that sodium dodecyl sulfate was what they responded most strongly to. And that he did cell ablation experiments and realized that that was because of the PHB sensory neurons and the PHAs that sense sodium dodecyl sulfate. Now, of course, White et al. Had done that amazing electron microscopy of the entire worm. And you can see here that PHB, this is just how we in neuroscience indicate synapses in what we call circuit diagrams. But PHB neurons form, sorry, I can't get my mouse back. Bad mouse, where are you? Maybe it's mad because I didn't use the model organism mouse. Okay, well, we'll see how far I can get without my mouse. But PHB neurons form synapses with AVA, which direct backward movement. And so AVA neurons, if you just stimulate them in any way, they will drive the worm backward. And it's because they form synapses with what are called motor neurons that form synapses with muscles that make the animal go back. And so when a worm backs up into a noxious liquid like SDS, PHB gets activated. And we actually think the synapses between PHB and AVA are inhibitory. I won't have time to tell you that story tonight, but trust me. And that will actually deactivate AVA, we think, which terminates that backward movement. Okay, so, ooh, there we go, got my mouse back. All right, so let me show you what that looks like. So this is a video I'm about to show you. This is a wild type or normal animal. It doesn't have any mutations in any of the genes that we're looking at. And this is its head. You can see it's a little paler here. And this is its tail. They're about 1 millimeter long. So this is actually an eyelash that we're going to tap it on the head with. That'll cause the worm to back up. And then we're going to use this little pipette here, this little glass pipette, to drop a little bit of SDS behind it. It'll absorb into the plate because the plate's dry. That's intentional. But we will circle that drop in red so you can see it. And you'll see that the worm almost immediately starts moving forward. It does not like that SDS, and it will terminate its backward movement and start going forward really quickly, okay? So la-di-da, worm going forward, we tap it with the hair, it goes backwards, and boom, it's out of there. Did not like the SDS. You probably didn't even see that, it was so fast. I'll show it to you one more time. La-di-da, moving along, worm pit goes back, boom, don't like SDS, I'm gonna go forward. However,
hey, how long is that total video?
Well, so usually I actually don't know, and I don't know.
Sorry.
Oh, here we go. There we go. That is 8.2 seconds.
Okay, so short.
Short,
yeah.
Absolutely. The average time that a wild type worm will back up into SDS is 0.5 seconds.
Got it. Thanks.
Okay, but if you have a mutant that has reduced synapses between PHB and AVA, like for example, UNC40, which I will tell you about in a little bit, then they will just keep backing into the SDS because the PHB will not be able to signal to the AVA to stop the backward motion. So here you can see, and the worm is a little bit smaller, UNC40 mutants are a little bit smaller, and it moves forward a little bit slower. I'll show you how we account for that down here, but you can still see a really clear difference, right? So it gets bumped on the head, starts backing up, there's your SDS, and la-di-da, no problem here, going right through that SDS, makes me happy. I could stay in it forever, right? So of course we don't want to just kind of say fast, slow. We like quantifying things in our lab. And so we will actually time 40 wild type animals responding to SDS and 40 wild type animals responding to buffer each day, and on the same day do 40 mutant animals and their response to SDS, and 40 additional mutant animals and their response to buffer. And so this is a really extreme example where the mutant animals respond to SDS they back into that SDS for just as long as buffer, just like this animal. And if that's the case, what we'll get is like 1.5 seconds divided by 1.6 seconds over the wild type average response to SDS that day, which would be around 0.5. Over the wild type buffer response is about 1.5. And so we get this response index of 300%. There will be a short quiz later, so I hope you're all taking notes. Actually, that's not true. All you need to know is that a high response index means the mutant responded slowly. And a low, as you can see, if the mutant responded just like wild type, if this was 0.5 over 1.5 divided by 0.5 over 1.5, it would be 100%, right? So 100% means it responded normally. Anything higher means it responded slow, and anything lower means it responded fast. Sorry, I'm enjoying, Walter, you should really be just sending your messages to everybody. They're hilarious. All right, so we wanted to really start to get at the molecular mechanism of this, like how the neurons don't just recognize each other. They're not sentient. They don't have eyeballs. They can't see, right? The only way they can really recognize each other on contact is if there are molecules that help them do this, right? And so my lab actually introduced this neuroligand grasp synaptic marker into a couple hundred molecules that we knew were expressed in these neurons. And what we do is we use genetic techniques to introduce loss of function mutations, which you can think of as deletions. They're usually not. Usually they're early stop codons that have the same effect as a deletion. But you can think of it as if we just got rid of that gene in that animal and we introduced this marker into the same animals, right? So now we can say, like, if wild type animals make this many approximately this many synapses, and we don't actually count them, we actually assess fluorescence intensity. And we do that with this image analysis software called NIH ImageJ. And more recently with this really amazing software that Phil Heller in computer science has developed for us called C. elegans, like SEE elegans. So Walter, there you go, another pun, courtesy of Phil Heller, which helps us assess the fluorescence intensity of these puncta. But you can just look at a picture and get the same sort of idea, right? So in a wild type animals, we see a normal level, but if we get rid of this gene called CLEAR1, we see reduced levels. And we can quantify that. We go from 100% in wild type to around 20% in CLEAR1. So then you're probably thinking, well, what does CLEAR1 encode? It encodes a receptor protein tyrosine phosphatase, which looks really similar to an immunoglobulin superfamily, one of those families of proteins that I told you had been shown to be involved in synaptic partner recognition. And the next molecule I'll tell you about, UNC40, the mammalian version is called deleted in colorectal cancer, or DCC, because it has an important role in colorectal and other cancers, actually is an Ig superfamily protein. And also, if you delete it, you have fewer synapses. Here you can see it's about 40%. And the next one I'll tell you about, UNC6, and the mammalian homolog is called netrin, is actually a secreted ligand that binds. UNC40 DCC. And again, if you get rid of it, you have fewer synapses. And so if you get rid of it and get fewer synapses, that means that the normal copy of it promotes synaptogenesis, or the formation of synapses. And so we geneticists frequently like to get rid of something and see what goes wrong, right? And in this case, it's synaptogenesis. I like to make the analogy when I'm teaching genetics, it's kind of like if you had a car, and I'm not the first one to make this analogy, but if you had a car and you were trying to figure out, and you were an alien from another planet, had no idea what cars did. And so you said, I'll just take off the steering wheel and then drive and see what I can't do. That's essentially it. You wouldn't be able to steering. You'd be like, right before you died, you'd be like, hey, I guess steering wheels are for letting you move the car left to right. Okay, so that's kind of what we're doing here, only in a way that's much less dangerous to students in the lab. So this was our first important discovery, that CLEAR1 receptor protein tyrosine phosphatase, UNC40 deleted in colorectal cancer, and UNC6 netrin are all required for synaptic partner recognition and that initial recognition and the formation of synaptic connections between neurons. So from both other people's studies and our studies, we now know that UNC40 DCC is expressed in PHB neurons. Those are the presynaptic sensory neurons, while CLEAR1 and UNC40, CLEAR1 RPTP and UNC40 netrin, why did I say UNC40 twice? UNC6 netrin are expressed in AVA neurons. And so we wanted to actually check whether expression in those neurons was sufficient to rescue their mutant phenotypes. And I could, this is actually a huge series of experiments. I'm just going to show you the ones for CLEAR1, but we did very similar ones to show that UNC40 in fact does function in PHB, and UNC6 does in fact function in AVA. And essentially what we did was to take CLEAR1 loss of function mutants where we had no functional copy of CLEAR1, and we made molecular constructs in which we took a promoter that drives expression in the AVA neurons that we're interested in, and we put that CLEAR1 coding sequence right behind it. And so now you're going to drive expression of CLEAR1, but only in AVA neurons, and the genomic copy is gone, so it's nowhere else in the worm. And you say, hey, can CLEAR1 function in AVA neurons? And the answer is yes. The synapses look very similar. We can quantify that. So if you look at this one, this is wild type animals have 100%, we normalize to 100%. Synaptic intensity, CLEAR1 mutants have about 20%. But if we then add back CLEAR1, but only in those AVA neurons where we know it's normally expressed, we can rescue the level of synaptogenesis. If we express it in PHB, not surprisingly, doesn't work. These are some other experiments we did just to show that the both the extracellular domain and the phosphatase domain are important. That probably only matters to the neuroscientists and cell biologists in the audience, but it will matter a lot to them, so I said it. And then we did it's such a great system where we can do the behavioral assays too, and show that synaptic function parallels what we see in grasp intensity. And so here, for example, we can see that wild type animals respond really quickly. CLEAR1 animals respond really slowly. If we then express CLEAR1 only in AVA neurons, the worms respond quickly again. We express it in PHB neurons, doesn't work. And similarly, if we delete the extracellular domain or make a mutation in the phosphatase domain, doesn't work either. So this was fairly strong evidence that CLEAR1 functions in AVA. And again, we did similar experiments for UNC6 to show that it functions in AVA, and UNC40 to show that it functions in PHB. And we can look even at the subcellular level to try and get an idea of what this looks like. We actually tagged the CLEAR1 cDNA, or the CLEAR1 coding region with YFP, a yellow fluorescent protein, and expressed it in AVA. And you can see this kind of punctate expression here that's concentrated in the posterior area where it forms most of the synapses. And we can do that with red. I'm looking at the time and realizing I should skip some things, but we can do that with other colors too, like M cherry, which is red. So if these molecules are driving recognition and synaptogenesis, then if we add more of them, we should be able to drive more synaptogenesis. And it turns out it works. So if we take wild type animals and we put in extra CLEAR1, just in AVA, we get more synapses. And you can see that here. Here are the wild type, and here are more synapses. And same for UNC6, it works. More synapses, yay. That does not work for UNC40. And I'll tell you why I think that is in a little bit. And similarly, I never expected this experiment to work. This is one of my students said we should do it. And I'm like, I'm not sure you can get the circuit to function faster. But they're like, well, because of the properties of C. elegans neurons, which I won't go into now, it's quite possible that they could. And so we did. And it turns out if you overexpress CLEAR1, the worms respond a little faster. If you overexpress UNC6, again, worms respond significantly faster. So it was really exciting to see these molecules really are drivers of synaptogenesis. We're starting to run a little low on time, so I will ask you to trust me that we did cool genetic experiments to show that these molecules all act together in the same pathway. We did both trans heterozygotes for the aficionados in the audience. Yes, Rachel and Bria, I mean you. And we can, if we make trans heterozygotes between CLEAR1 and UNC40 or CLEAR1 and UNC6, we get reduced synaptogenesis, which actually indicates they're in the same pathway. And we can do similar things with double mutants. We can use UNC6 overexpression to do epistasis. Well, at least the closest to epistasis we can come, and show that CLEAR1 is likely acting downstream of those two molecules and that they act in the same pathway. And everyone other than Rachel and Bree, trust me, that's what these show. So that leads us to this model where we have our presynaptic neuron, PHB, and our postsynaptic neuron, AVA here. And UNC6 netrin is secreted from AVA, and it probably binds to its canonical receptor UNC40 DCC in the presynaptic neuron, and then signals to CLEAR1 in the postsynaptic neuron. And this interaction triggers synaptogenesis between these two correct partners. Now, if we overexpress UNC6 or CLEAR1, we can get more synapses. But if we overexpress UNC40, we don't, which tells us that probably there's ample UNC40 already there. And really what's limiting here is the UNC6 netrin and the CLEAR1 RPTP. Okay, so that was really exciting to find this new mechanism for these highly conserved molecules that have roles in cancer and schizophrenia, and realize that they have this new role in recognizing and promoting synaptogenesis between correctors. So the second part of my talk, which I promise will go a little faster, was about how activity can determine the level of synaptogenesis. So if we have activity through the neuron, a lot of it, that can promote additional synaptogenesis. And this is something we've known in mammals for a long time, although there are still huge gaps in our understanding of this at the molecular level. And that's potentially something we can address in a simpler model organism like C. elegans. But for the most part, we've thought that C. elegans had pretty determined synapses, right? That every animal was exactly the same. So we were honestly a little bit surprised when we were doing the studies, and we noticed that L1s really didn't seem to have much fluorescence intensity compared to L4s. Here's the quantification of that. L1s low, L4s high. Ugh, I didn't tell you what L1s and L4s are. Worms have 4 larval stages. When they hatch, they are immediately L1 or larval stage 1. Then after about a day, they molt and become L2, and then they molt and become L3, and then they molt and become L4, and then they molt and finally they become young adults. So anyway, L1 means right after they hatched. We assay them within an hour of hatching. And L4 is an almost adult animal. So we decided to go do a time course, and these are single animals here. This is one animal and another animal and another animal and another animal and another animal. And the first larval stage, second, third, fourth. And we noticed this amazing thing, which is it's not just growing proportionately with the length of the animal, right? Which is increasing during this time, but at a fairly steady rate. In fact, you see this huge increase from L1 to L2, and then it just kind of stays the same to L3. And it actually goes down a little bit before L4. And I'm not showing the picture here because once you've anesthetized animals 4 times in just a couple days, they don't look so good as young adults, but it bounces up after that. And these are all the individual traces, and it's not shown up really well here, but this is the median of those traces, showing exactly what we're seeing here, that it starts low, goes up high in L2, stays high, and then comes down. And we can do population assays to get to image worms that haven't been pre-imaged. We just image a bunch of L1s and a bunch of L2s, bunch of L3s, bunch of L4s, and a bunch of young adults. And we see exactly the same pattern. It has this huge burst of synaptogenesis at L2. And so we thought, well, wouldn't it be cool if that was actually activity-dependent synaptic regulation, like we see in mammals? What if we could actually study it in this organism? And so we thought, what could they be sensing? There's no SDS in the plate. So my students literally looked at every ingredient of that are in our plates, and they found out sulfate was the answer. They don't sense sulfate as strongly as sodium dodecyl sulfate, but they sense it significantly more than anything else in the media. And so this is showing you that again. So we decided to look in mutants that have no sensation at the tail. And there are these key 3 mutants that have been thoroughly studied by others. They have defects in the very tips of their dendrites called the cilia that keep taking in information from the external environment. And if we look in these, they look about the same in L1s, but when we look at L4s much later on in development, they have very few synapses in comparison. And we can quantify that here. Again, just like you'd expect, right after hatching, they look pretty much the same. But then we look at L4s, and the key 3 mutants really have way fewer synapses than these wild types. We thought, well, if it's really the sulfate, then if we add more sulfate, we should get more synapses. And it worked. When we added sulfate over the developmental period, we actually got synapses. And you can see that here quantified significantly more. So we thought, wow, there's activity-dependent synaptic regulation in synapses. It's really exciting. And we wanted to know at the molecular level how it worked. Well, I have to quickly digress to tell you how we got to the molecular level, because if we wanted to look at activity, we needed to understand what the circuit was really sensing. It turns out there's no SDS in the environment. It's human-made. So we wanted to figure out really what is this circuit sensing? And through long experiments that I will not tell you all the details of, it's Streptomyces bacteria. So Streptomyces bacteria are actually, I mean, no offense to the microbiologists in the audience, but they are really mean little bacteria. They secrete nematicides, chemicals that kill worms. So if you are a worm, you do not want to be near Streptomyces because it will kill you. And we put little drops of Streptomyces back behind it, and it turns out, yeah, they will stop backing up into that just as quickly as they stop backing up into SDS. So again, remember the long response, a slow response, which they do to saline buffer or control E. coli or Bacillus, but they have a really quick response to SDS. Streptomyces avermatilus, Streptomyces costa-ricanus, Streptomyces milbemycinicus. Okay, microbiologists, you just have to be proud of me for saying all of that correctly all in a row. And we were interested whether it was actually the bacteria or something they were secreting. And so we made cell-free supernatants, and they respond to them exactly the same. So we think it's a secretion. And pretty short, what are they secreting? Dodecanoate. It looks a lot like SDS. And actually, it was my brilliant collaborator, Laura Miller-Conrad, who first thought of this. She's like, you know what is biologically relevant that looks a lot like SDS? And we went through lots and lots of testing, everything. And she actually, and her collaborator at UCSF did these beautiful experiments where they actually looked at the secretion products and showed that streptomyces decanoate. And we tested it, and sure enough, they're responding to it. Not quite as quickly as SDS, but very close. So we finally had an actually relevant stimulus for the circuit that we would expect to find an actual G protein, an actual receptor for it, because that's what the circuit does. So that was really exciting. Oh, there's lots in the chat. Okay, sorry, you guys already caught up on these, I'm guessing, but Cheryl, you're awesome. Okay. So the SRB6 GPCR, long story short, we tested like every G protein coupled receptor. Oh, I should back up and tell you what a G protein coupled receptor is. So odors and many other chemical cues, like in liquids, turn out to be sensed in pretty much all organisms by G protein coupled receptors. There are other families like TRP family ones that are very important, but the G protein coupled receptor family is a huge family. It's in fact one of the biggest families of proteins in humans. And they're conserved all the way down to C. elegans, that family. And long story short, I won't walk you through it, but this particular one out of all the ones we screened, called SRB6 for no reason I need to describe, is required to sense streptomyces and dodecanoate. And it functions in PHB neurons. We can do those same types of cell-specific rescue experiments where we only express it in PHB neurons, and we can completely rescue the function of the PCR just by putting it in those neurons. So we're pretty sure that SRB6 is functioning in PHB neurons to mediate sensation of streptomyces, of dodecanoate, which I'm from now on going to call lauric acid because it's really hard to say dodecanoate, and luckily chemists give you multiple options of what to call almost every chemical. So from now on, I will call dodecanoate lauric acid. So long story short, SRB6, in addition, this is our control, is responsible for sensing lauric acid, right? Key 3 mutants don't sense it because they don't have proper cilia. 3 different mutant alleles of SRB6 also fail to sense it. With sulfate, we also see a defect. So key 3 mutants really don't sense sulfate at all. SRB6 mutants definitely have a defect, but it's not quite as strong. So there may be another GPCR that's working as well, but most of this sensation of sulfate is coming from SRB6. And so we wanted to know, can we actually link an individual GPCR to this sensory signaling dependent post-embryonic synaptogenesis? And the answer is yes. And it's kind of even cooler than that, because if you look at the wild type animals, you have this pattern we talked about, this jump from L1, L2, then goes down to L4 and back up to young adult. It's so cool. You see the same thing, but it's all lower. It's all at about half the level. So you can see like there's some genetic regulation in there, and then superimposed to this activity dependent regulation of the magnitude, which is just really exciting. We haven't really gotten this type of resolution in another system. So let me try and summarize. So we've been able to find this mechanism of conserved molecules that are linked to neurological disorders, a new mechanism that wasn't known previously that regulates recognition and synaptogenesis between neuronal partners that generates a functionally relevant circuit. And we've been able to determine that, in fact, activity can determine synaptic levels in C. elegans like in humans. And we've gotten this resolution into understanding exactly how it's doing it during the lifespan of a worm, that activity is promoting this burst of synaptogenesis with its already recognized partner. And we've been able to identify the ecological, the relevance of this circuit in helping the worm avoid this toxic bacteria, and identified the specific receptor that senses the secretion from the worms, and found that this receptor is actually involved and required for this activity dependent super imposition of activity on regulating the magnitude of synapses. And we can bring that back to really thinking about why that's important, because it's going to, the genetics are going to tell you in general how the connectivity forms, exactly who's going to form synapses with who, what circuits will happen, and what the animal will be able to do. But then activity or nurture will then come in and help each individual reach a level that's appropriate for its environment and respond to its environment appropriately. So of course, we're looking into this more. My students have a million projects I haven't told you about, but we're building out these pathways now. We're trying to understand them, trying to understand other aspects of development. It's a really exciting time. And I want to thank all the amazing lab members who I work with all the time. This top row is my graduate students and my lab managers, who are all amazing. These are my senior undergrads here, who all are helping to train and doing amazing experiments. And this row here is my amazing new cohort for fall, who are all getting trained and all doing amazing things. I also need to thank, I have so many past lab members. I have not listed them all here, but I've listed a bunch of them who made amazing and important contributions, most of whom are in grad school or medical school now, or pharmacy school, or have graduated from such, or working in biotech. I also want to thank my amazing collaborators. I didn't mention this, but Martina Bremer in math, I've collaborated with her on everything I've ever done. And it's an amazing collaboration, because she helps me know that that we really know what we think we know. And that's the most important thing. Laura Miller-Conrad, I told you about the amazing stuff she did. Daryl Eggers, who worked with me on figuring out that the tail parts that I had to leave out, but that the tail of SDS was important, that the head group was important, and pointing me toward Laura. Sammy Curry in computer science did some work I didn't get to show you here. My collaborators at UCSF, Noelle Latoile, Bo Huang, Saul Cato, and then our amazing technical staff here in the biology department and the College of Science, who've done, you know, we couldn't do anything here without them. They are the glue that holds us all together. So I'm very grateful to them. And of course, my funding, because you can't do anything without that. So any questions?
Thanks, Miri. I got one. I always have a question.
Awesome.
It may not be a good question. So you seem to have discovered a way to cause synaptogenesis. In theory, you have the tools to do this.
We do.
So, so I'm imagining a Frankenstein scenario where someone says, "I want to be super genius."
We're just after Halloween. This is an appropriate time to do so.
So, so I'm going to create a potion that will increase the synaptogenesis in the brain of me or my kid or something like that. Mm-hmm. What's wrong with my horror story?
Well, you are completely correct that it would be a horror story. So it's very important that we make the right connections, not just tons of connections everywhere, right? If PHB had formed synapses with any of those other gray neurites I showed you, it wouldn't have functioned, right? It has a limited, other experiments we've done indicate it has a limited number of synapses it can form. And if it doesn't form them with the right partner, then it won't be functional. So yes, you could potentially express these molecules and drive synaptogenesis throughout the brain, but it wouldn't be a good thing because it really matters that you have intricate, correctly formed synapses.
Thanks. All right, first question or first hand up is from John Mark. John Mark.
Hey, John Mark.
Hello, how you doing? Awesome
Good.
talk.
Oh, thanks.
Hey, I'm so cool with all right. So I was following along when you mentioned that the long nonpolar group with the sulfate group on the end, that it doesn't like that. And I thought, okay, well, I bet there's some protein that overlaps with the sense of smell. And then you said that, I was like, okay, cool. And then I was like, but then you said that it had a similar response to the one with the carboxylic acid group with the same fatty tail. And that threw me off a little bit because as far as I mean, I don't know much about this at all, but I thought there was a high correlation with like vibrational modes and molecules and how we perceive their smell. Like, I think like there's like people who do machine learning models, and that tends to be one of the best inputs to give it is like spectra of vibrational modes. And then that predicts how something's going to smell. So has anybody looked into this? Like, because it seems like that's just so much more mass that it seems like it would have a very different, I mean, I don't know, I guess you can't ask them if it tastes the same or I don't know anything about this scent that they don't like this chemical. I don't know how to think about that. But.
No. I think this is a really great question. Okay, so the vibrational stuff I know less about. I know enough to nod when you say it because I've heard it, but I don't feel like I'm expert enough to comment on. But the way we generally think about G protein coupled receptors and their binding to odors and chemicals is that there are pockets. There are binding pockets in the extracellular domain in the 3D structure of the protein. And that these pockets are very specific and they're like formed to the molecules that they sense, and that they, a group that has like if it's meant to bind like a positively charged area, you'll have like charges around there. If you have a hydrophobic tail, there'll be like hydrophobic amino acids, etc. So we do not have a crystal structure or even a good predicted structure. And this is probably something I should talk to you about for SRV6. I would love to have that. But what we imagine is the case is there's this binding pocket is for, fits perfectly for the head and the tail. And then just the head alone, which is the sulfate, can sit in there and activate it to a certain extent, but not entirely. And you really need that tail to activate it entirely, right? So if you get rid of the tail, it'll activate a little bit, but not all the way. If you get rid of the head, like the tail alone almost doesn't activate it at all. I didn't show you that data, but we did publish it.
Cool. Well,
Yeah.
thanks. That's really neat stuff.
I'll talk to you more about that.
Please do. Yeah, I'd love to.
So I know early on in the talk, Walter had a question, and let's see.
I refuse to answer any questions of Walter's unless they begin with a pun.
That's not, I literally have used like so many puns, like, oh gosh.
You could just read the one you just put up.
Yeah, now I'm definitely, this is getting on my nerves. And
Nicely done.
This was a nice wormtail. I don't know.
I can cut off your talking at any time, Walter.
The speaker asked me to do it. My chair is saying going to encourage me. The dean is going to come. All right. So I was curious early on in the talk about the SDS experiments. One, I felt like very bad for the worm, but then I also was wondering, does the SDS damage the worm's neural circuitry at all? Because I mean, it went all the way in there, and I mean, does that have any downstream effects for its movement or anything, or is it just uncomfortable?
Well, because we're worried about that, we never test the same worm twice. So once we have tested a worm, unfortunately, that worm is not long for this world, and we move on to the next worm. However, I could tell you, For a while, so there are these experiments called adaptation experiments where you actually like incubate an animal in an odor that it senses, usually not SDS. In fact, nobody's done it with SDS other than us. And it turns out like you put just some small molecule that they respond to that they like. And if you incubate them in it for a long time without food and then ask them whether they're going to move toward it or not, they won't anymore because they no longer associate it with food. Right. And so we tried doing that with SDS just to see what would happen. Like if we, incubate it, and it's a really low concentration, like we put it below the critical micelle concentration. So it's not actually dissolving membrane or anything like that. And that was Daryl's idea, by the way, to start doing that because we were just barely over the critical micelle concentration before I started working with Daryl. Thank you, Daryl. So we incubated it in SDS, and even after like a while of incubating it and putting it out, it can still respond to SDS. It does not adapt to SDS. So at least at the concentrations of SDS that we use, it's not harming the worm. But definitely if you used higher concentrations, yeah, right, because SDS is a detergent at high concentrations.
Okay, great, thanks. And also thank you for including bacteria in your talk. That's really important.
No problem. Did it for you, Walter.
All right, do we have any other questions from the audience? Oh, Sarah Smith has raised her hand. Sarah, go ahead.
Hi, can you hear me?
Yep.
Yes. Okay, awesome. Very great talk. It's really fun to learn more about your work.
Oh, thanks, Sarah.
I had just a kind of a methodological question. How do you make the mutants? Like, what's the process that goes into them and how long does it take? Like, is it something that a student can do in a semester?
Yeah, okay, so there are different ways and it depends on what you want to do. You can use CRISPR. If you want to make a very specific mutation, like you want to delete a gene, or you want to make a very particular mutation, like you already know what you're interested in, you can use CRISPR. And could you do that theoretically in a semester? Yes. It's taking us a little bit longer. We're doing some CRISPR modifications just because it's our first time. But once we get up to speed, like, will we be able to do it in a semester? Yes, but it would pretty much the student was doing a semester. You can also mutate genes randomly. Don't know what you're looking for. You can just make random mutations throughout the genome and then just look at a bunch of different worms until you find an interesting phenotype and then go back and map the mutation. And that is something you can do in a semester. And we have done screens, they're called screens, forward genetic screens. We have done screens in the lab. In fact, one of them pulled out clear one.
Cool. And do you use like antisense or RNAi for mutations that you don't want to be too deleterious?
Yeah, so I didn't talk about it here. In another project, we have used cell-specific RNAi to knock out, to knock down expression of a gene in a single neuron. And yeah, we can absolutely do that in C. elegans. We're actually doing that in the project. We're trying to figure out the receptors, the neurotransmitter receptors in our circuit because it's not known, and my students have, I think, figured it out, but since we haven't done the final experiments, I'll knock on wood when I say that, but we're doing cell-specific RNAi of each of those receptors to really show its functioning in AVA neurons where we think it is. And it's a great
so
technique.
Awesome. Yeah, it's so awesome the elegant science that can be done in these models. I get a little jealous.
Thanks. And I see what you did there with that elegant science and
Thank you.
Thanks, Sarah.
All right, we got one more hand. It's Steve Brands.
Oh, Steve.
Go ahead, Steve.
Hi. Yeah, I was just typing out a long question, but I can just say it instead.
Cool.
And it was sort of prompted when you said that that you don't let, once you've tested the worms, you don't let them live, which seems really, cruel, but
I know, it's terrible. We feel bad. Okay, to be fair, when we've got them on the microscope, we can rescue them, but once we've tested them for behavior, yeah, we don't rescue them.
So here's my question. At least my understanding of the way we have memory and learning in humans is that, if you use some neural pathway if you practice your times tables over and over that gets strengthened, right? And if you stop using that neural pathway, it stops being so active. So if you had let the worms live, would the synaptogenesis, would it reverse itself?
Interesting. So, I mean, when we put the worms on the plates with the additional sulfate, which they also sense, so that throughout their development, they're sensing sulfate, it does exactly that, right? It builds more synapses. And I showed you that experiment early on, right? To show that these synapses were activity dependent, right? And that is related to learning in a very kind of simple way, right? You could think that the worm is learning to respond more quickly to sulfate, which it will if it has more synapses, right? Because there's a lot of sulfate in its environment, right?
But but the question is, does it unlearn, if you don't use that, if you
Right.
don't keep sulfate in the medium and you just let it live a happy, life as a senior?
So I think that's a really, I think that's a really great question, right? Could we take those worms, remove most of the sulfate from the media? They can't live without any sulfate, but remove most of the sulfate from the media and say, would that genesis go down over time? I think it probably would. We haven't done it, but that would be really interesting to do. We actually have an entire other side of the lab that's devoted to understanding learning and memory in worms and understanding the role of sleep in learning. That's a big collaboration with UCSF. And I did not have time to talk about that today, but a lot of what you said, if you come to happy hour, like, 15 more minutes of thoughts on your question that I'd love to share with you.
Okay.
Okay, well, I want to thank Miri for a great talk, and also want to respect people's time. We're at a 5:15, so that's all right. That's all right.
Well,
We'll talk about it later. But this is really great, and I hope that many of you are able to join us for happy hour afterwards. Thanks
I'm
again.
looking forward to your talk, Michael.
I'm working on my slides right now.
Thanks, everybody.
Thank you, everyone. All right, see you at happy hour.
All right, see you soon.